Okay, I will slowly get started. If you're in any of the other rooms and you think I'm not audible, please write something in the chat so I can adjust the settings. I'm reading it on my phone, so I can still read it when I'm doing something else. Okay, so for today we have two lectures, and of course we have a practical. The first lecture is on study design, which is more or less like the video that we posted before. And the second one will be from Harold. So we switched the topics around from Wednesday and Thursday. First, he was going to have a lecture on data manipulation, visualization, and subsetting, stuff like that. So the basic tools for working with data. But yeah, my lecture is already finished, so we're going to start with this one. And then we're going to do only the visualization part from Harold's lecture, which I think will nicely integrate with the practical, which is drawing in base R. So I had a question from a student 1 or 2 days ago about using other packages during the graded assignments whatsoever. I have no problems with that. You can use whatever as long as it works. But for this specific assignment, you cannot. This assignment, you have to draw something using only what comes pre-installed with R. Yeah? So I will tell more about it once we get to the practical, but I just wanted to quickly give an overview of today. Okay, I don't see any questions, so I think I'm going to assume the audio is okay and get started with the lecture. So the topic of today is study design. So this course, Advanced Statistics, is of course important because you have to be able to judge other people's works, and for that you need to have some idea of the method they use. Even if you don't understand the specifics of it, you need to be able to say whether you agree or don't agree with the methods they use given the study design they have. And also, of course, once you get to your internship, a lot of you will have the freedom to make your own little experiments or make decisions within the context of your internship. And also, anyone pursuing an academic career, study design is an integral part of being a scientist. Now, there are a lot of statistical aspects of study design that I want to talk about today. But first, let's define what study design is. Study design is, for a statistician, it is maximizing the power of your study, maximizing the translational value of your study, and simultaneously minimizing the bias. Yeah? And to do that, you have to define a clear outcome. You have to take certain considerations to minimize confounding and bias. And power I will talk about later in the lecture. Okay, so what you would ideally do is just collect infinitely many independent observations and then measure everything about those observations that could possibly affect the outcome, and then run some incredibly large model that will estimate every independent effect. But of course, in reality, you're limited by the time investment you can afford. Research costs money. There are ethical concerns, especially if you're working with live subjects. Then there's of course privacy concerns and security concerns, and any other constraints you can think of means that we have to design a study such that it is maximally efficient. Yeah, so we want to make optimal use of resources. But there's more to it, because the better your study is designed, usually the easier the analysis is. So if a study is really well designed, sometimes it all boils down to one t-test and you have a valid answer to your study. And of course it increases the translational value, and what I mean by that is that if your study is well designed, if you pick your samples in a smart way, then you can generalize the results to a much larger group than if there are certain flaws or quirks in your study design. Okay, for the purpose of this lecture, this one is both part of basic statistics and advanced statistics. I'm only going to talk about study design in the context of hypothesis testing. So that is essentially any research question that you can reduce to a yes or no question. You could design a hypothesis test to answer it. So you could think of, are men taller than women? Are bees more attracted to certain colors of flowers than other colors of flowers? And does a certain antibiotic like tetracycline work as an antibiotic against a certain bacteria like E. coli? Yeah, but of course you cannot reduce everything to a yes or no, and that would be stupid. You would really limit yourself as a scientist. So yeah, think of which patient should receive certain treatment. If you're going to do it with a hypothesis test, you're going to introduce a really large multiple testing problem. It's also going to be really inefficient, and you are optimizing a model for inference when what you really need is a predictive model. Also, let's say you want to estimate the time until the next large earthquake in a certain area happens. That's just not something that you can meaningfully do with a hypothesis test. Or what is the weather like tomorrow? These are all questions that you can answer with statistical models or predictive models, but not with hypothesis tests. Yeah, so in the second or third, okay, anyway, during the mixed models lectures of the advanced statistics course, we're going to come back to study design, introduce a couple of other elements that allow you to apply to a little bit more than just hypothesis testing. But for the purpose of this lecture, the only thing you really need to know is what a t-test and what a chi-square test is. And in case you don't, t-test is a comparison of means, and a chi-square test is a comparison of frequencies. And that's enough to follow this lecture. Okay, so first term I introduced is bias. We want to minimize the bias. So what is bias? Well, you're going to draw a sample, and each individual observation you draw from the population of interest is going to differ to some extent from the rest of the observations. And that's fine. But we want the observations that we draw to be, on average, equal to the population of interest. Yeah, and if there is a systematic deviation from that, that is what we call bias. So a quick example, let's say that you have two types of plates. So you're doing an experiment and you want to say which of these plates, like let's say they have a different medium, is better at growing bacteria. Well, then you have 4 plates of one group, 4 plates of the other group, for example, and you're like, well, I want to speed things up, so I'm going to use 2 ovens, and we're going to put all of the samples A in one oven and all of the samples B in another oven. This is, of course, a stupid idea, because any difference in heat transfer between those ovens will now be a systematic difference between A and B. So you can no longer attribute any difference you find to the groups, because you don't know if it's because of the groups or because of the ovens or because of both. There's no way to to extract the difference of interest. Yeah, so this is an obvious example, and if one oven is very old and the other is very new, then this difference is going to be large. But in general, you are dealing with things that are much more subtle, yeah? And it might not always seem like it's going to be a big problem, but for the purpose of study design, always try and prevent this type of situation. So this will bias the difference between A and B. A solution is very simple, randomization. So if you randomly assign both sets of plates to both ovens, then there is still a systematic difference between ovens. But now that systematic difference is averaged out across groups because you randomly assign them to it. Yeah, so these 2 Bs might be slightly better grown than these 2 Bs, but then the average of that will be 0. Yeah, that's the idea of randomization. So randomization averages out bias. Okay, so bias also applies to sampling. So let's say that you're conducting a survey, you have some population of interest, you want to say, what do people in the Netherlands think of this political statement? And then what you want is some random representative sample of the population of interest. If you say, okay, I'm just gonna ask this my friends and my family to fill in this survey because it's easier, then that is kind of like this situation. It's only drawing a sample locally to those close to you, and those close to you will probably on average be different than the entire population. Yeah, so you are introducing a bias by doing that. If I hand out a survey over here, then I will have a pretty good idea of what first or maybe second year master students at the IBL think of whatever my survey is about. But I cannot generalize those conclusions to all biologists in the Netherlands, or let alone all people in the Netherlands. Yeah, so it's really important that you define what is the population of interest. Yeah, so is it biology students from Leiden University? Is it master students, master biology students from Leiden University? Or do I want to talk about all students in the Netherlands? Or do I want to talk about all students in the world or all people in the world? and then you have to somehow design your study such that you can actually draw a conclusion about that entire group. Yeah? And well, you can already see that this is going to be a much more complex effort when you want to draw conclusions about the entire human population, because you somehow have to involve research groups from all over the world that somehow are going to pick random people in their country. So this is incredibly difficult. This is also what, for example, makes coronavirus research very difficult. Okay, minimizing bias is not that difficult. I'd say the most difficult part is correctly determining your population of interest. And the previous example, I talk about a literal population, right? I talk about the human population. But in general, in statistics, the population means the source from which your samples came. So when you are putting bacteria on a plate, the population would be all bacteria of that kind. Yeah, and how you are going to design your study around it, that's the difficult part. The not so difficult part is preventing bias from potential systematic differences, because what you can simply do is use random sampling and random treatment allocation, and then there will still be bias, but on average it will be 0. Yeah, randomizing in R is very simple. If you only have, let's say you have 10 places to distribute, just say sample and then the numbers 1 to 10, and then this is the order in which you're going to treat or assign or deal with your samples. Yeah, and for example, if you have 2 ovens, then you could say, okay, I'm going to put samples 1, 10, 9, 2, and 7 in one oven, and we're going to put the other ones in another oven. And you can still use 2 ovens, you can still speed things up, but now there will not be a systematic difference in groups. Of course, there are more complex study designs, like maybe you have a continuous variable, then this is a bit more complex. But I have written about this in the first book, Introduction to Biostatistics, which you can find on GitHub, paragraph 3.6. Okay, then the next term is statistical power. That's the thing we want to maximize, because statistical power is your ability to detect a difference in your sample if there is a difference in your population of interest. And the thing with any kind of empirical research means research where your conclusions are dependent on observations or experiments. And any science where we do that, like biology, chemistry, applied physics, then a sample, you always have to keep in mind the results of your sample are by chance. So it's not the whole population, it always comes with uncertainty. That has a really big implication for how you should write about empirical science, because you should talk in terms of evidence and not in terms of proof. Yeah, so for the Dutch people around here, we call both of those bewijs, which is really annoying when writing subtitles for this video. But evidence would be bewijs or aannemelijkheid, and proof would be onomstotelijk bewijs. Yeah, and the latter, proof, is something that we do not have in empirical science. Yeah, we are just making things more and more and more likely, but we don't prove something to be true. Okay, so what's the difference? This is the Pythagorean Theorem. I'm sure most of you have seen this before. It's a squared plus b squared is c squared for any right triangle. Yeah, so that means that if I square, the length of A and I square the length of B, then the total surface area will always be equal to if I square the length of C. Yeah, and I can pick any values for A and B and C, and this will still hold. So this is irrefutable. There are no counterexamples to this theorem. So that's proof. Yeah, but in the case of evidence, this is Bob. He is near the cat. The cat is dead, and no one else is there. Then maybe he killed the cat, but there's always some other possible explanation. Yeah, maybe he did kill the cat, or maybe the cat ate poison, or maybe he got run over by a car, or just randomly got a heart attack. We don't know. We can't ask him now. So maybe Bob just happens to be in the wrong place at the wrong time. And it's a bit of a ridiculous example, but the point is that you can never be 100% sure about something in empirical science. Yeah? Same goes when sampling. Every sample will slightly differ from the next. So don't give too much value to whatever p-value or whatever conclusion is at the end of your research. Always take it into context. You know, how many samples did you draw? How did you draw your samples? Is that unbiased? And what is the population that you can actually generalize those results to? Yeah? So just always keep in mind that what you're doing is collecting evidence and not proof. Yeah? And that your sample is just one of many possible subsets of the population. Yes?
Is it also the case when you measure the complete population?
Okay, so the question here is this also the case when you measure the complete population? That's a very tricky question, because to answer it, I have to explain the difference between a biological population and a statistical population. So if somehow, I don't know how, given politics and other reasons, we could measure every individual person in the world at a given moment, then you could generalize the results to, of course, the entire population at that particular moment. But 1 moment later, some people will have died, some people will have been born. The biological population, you could measure it completely at a given time in theory. In practice you can't. The statistical population is more like the source from which all human beings come, so to speak. So that means that you would have to measure everyone who is, everyone who has been, and everyone who could have been, and everyone who will be. And that is something that I don't think we can do. So because of that, there is never quite proof, even if you measure the entire biological population. Yeah? Okay, so that brings me to the statistical way to think about this. You can make errors, right? Your sample can say there is no difference, even though there is a difference in the population. And the other way around, your sample can say that there is a difference, even though on average those groups are the same in the population. Now, here's an example with a test for a viral infection. So there is the ground truth. Either you are infected or you are not infected. And the test is either going to say that you have the virus or you don't have the virus. Now, when a test says that you have the virus, even though you don't, that is called a false positive. And the other way around, when you are infected and somehow you slip through and the test says, no, you're good, that would be a false negative. And these things, we can reduce them, we can try and control the chance that they happen, but you cannot make them go to zero. They always have a chance of occurring because we don't have the whole population. Okay, so the way to express that is like this. We call the error terms alpha and beta. So alpha would be the type 1 error or the false positive. So saying that there is a difference when there isn't, saying that there's an effect when there isn't. And beta is the other error. That's when you fail to conclude a difference even though there is one. Now, that finally brings us to power, which is 1 minus beta. Yeah, so it's 1 minus the chance that you would fail to detect a real difference. Because that is your chance that you are able to pick up the difference that's there. Yeah, and that's what we want. We want to be able to have that power. Okay, so alpha is something we choose. Yeah, we can be more or less strict about the chance of a false positive. In biology and many fields, it's common to say 0.05. That means that you have a 5% or 1 in 20 chance of concluding a significant difference, even though there is no difference in the population. Because it's a sample. Yeah, it's just a random subset. It could coincidentally have a significant difference. And that chance is alpha. We choose it. Beta we don't choose. There is no way to do that. But we can influence beta. And that's what we're going to talk about now. How can you design your study to have sufficient power? Okay, so in essence, you need enough power to demonstrate a difference that's there with your sample. The way to increase power, the easiest way is to just get more independent observations, right? But it's not always an option, but that's what we want. Ideally, the more the merrier, collect as many observations as possible. Okay, but the question, of course, is how many do we need? Because practically, we cannot collect infinitely many. So there are some things that influence it. I'm first going to give you a vague overview, like what is more or less going to influence your power. Then for the t-test, I can express it exactly. There's an exact relationship. And we're going to do more than just t-test and chi-square test in this course. So then I'm going to go back to vague, where I say, okay, here's how you could more or less figure it out. But there's no exact relationship for complex analyses. Okay, so if the difference of interest is small, there's a subtle difference, then you need a very large sample to demonstrate that difference with significance, right? And if the difference is really large, then even in a small sample, you'll probably see it. Similarly, the variance, so that's the extent to which individual observations differ from each other, if that's really large, then there's going to be really large overlap between groups. And it's going to be more difficult to say, okay, yeah, sure, people differ from each other. Everyone differs from each other, but on average, this group is different from that group. You would need more observations if the variance is large. Okay, now finally, if you're very strict about what counts as significant, so if you're very worried about false positives, yeah, you can set alpha very low, but that also means that you have a smaller chance of concluding something is significant because you set the threshold very low. Yeah? So yeah, number one, the difference of interest, we can guess. Number two, the variance of a system is inherent. It's not really something we can influence. There are aspects of it which we can influence, which I'll talk about next. And finally, alpha you can choose. Yeah, you can just go with 0.05, but you should think about it. You should think, is that a reasonable choice within the context of my research? If you have to decide, okay, again, I wouldn't use a hypothesis test for this, but for the sake of argument, if I have to decide who will get chemotherapy, then a 5% chance to treat someone with chemotherapy when they don't have cancer is ridiculous, right? That is way too high. But on the other hand, if this is a pilot study and I just want to know, okay, is this bacteria, does it form larger colonies than this one? Who cares if you're wrong, right? It's just a pilot study, we're just trying to figure it out. Go with something higher, like 0.1. Yeah? So you can choose it yourself, as long as you declare it before you start collecting observations. Okay, then some practical considerations. So this would be the order in which you set up an experiment for hypothesis testing. The first thing and a very important step is to define a clear primary outcome. You should not collect any data if you're not quite sure yet what you're going to do with that data. That's my point. So let's go over the three examples again. Are men taller than women? Then your primary outcome is the height in centimeters or in inches, whatever, of individuals. Yeah, if you want to know, are bees more attracted to certain colors of flowers than others, then you would count the number of bees that visit a flower within a given amount of time, yeah, at a given moment on a day. Okay, and then the last one, does tetracycline work against E. coli? That kind of depends. Depends how strong tetracycline is. If it's really, really strong, it kills off everything, then yeah, we can count colonies, but we're going to count 0 or not 0. So in that case, you should probably have multiple plates and see what percentage of the plates or what proportion of the plates did not grow colonies. Yeah, but if it's a milder antibiotic and E. coli can still grow but very limited, then you could count the number of colonies per plate and have plates with and plates without tetracycline. Yeah, so that's the primary outcome. Then using that primary outcome, you should convert the research question to something very, very precise, something that can be expressed in math, which is a testable hypothesis. So yeah, first we have the research question, are men taller than women? It's too vague. So because what do you mean? Do you mean that all men are taller than all women? Or do you mean that men have a larger maximum height than women? So you have to be precise, right? So in the case of a t-test, I would say that the average height of men, average, that's the important part, is larger than the average height of women. And now it's precise. Now it's two measures that we can compare. Second one, you could say that bees are, again, on average, more attracted to certain colors of flowers than other colors of flowers. Of course, you'd have to define what those colors are. Third one, yeah, you'd have to choose a concentration and then say, using this concentration of tetracycline results in a different number of E. coli colonies on one plate compared to a control medium. Yeah? Okay, so there's still a problem with these statements, and that's that, yeah, whether this is true or false kind of depends on what do we mean by larger, right? larger. And what do we mean by more? And what do we mean by different, right? It's still not quite something you can express in numbers until you agree on it. Well, we can circumvent that. We can just instead agree on accepted chance of false positive and say, is it significantly non-zero? And that is what you do with the null hypothesis, yeah? So in the case of the t-test, you would say that the mean of men is less than or equal to that of women. Okay, why this? Why not just equal? Because, you know, I may already have an idea that I think men are taller than women on average, and I only want to test in one direction. And if this statement is not true, then men must be taller than women, whatever taller means, right? So that's why the statement is like this. Second one, you could say that the number of visiting bees per flower is equal for each of the colors that you consider. Yeah, and if it's not equal, then we'll reject this null hypothesis. Last one, again, depends on the strength of the antibiotic. In the first example, if it's really strong, then you would just say, okay, the proportion of plates with colonies is independent of treatment. So you can make it like a contingency table, right? You have treated, not treated, and you have with colonies, without colonies. You count both, perform a chi-square test, and you're done. Or you could use the number of colonies on tetracycline and control plates and state that they're equal. Yeah, if it's not a very strong antibiotic, you could even do this experiment with just 2 plates and count the number on one plate and the number of on the other plate, and then see if that differs from a 1-to-1 ratio. Yeah, so that's the null hypothesis. First you define a primary outcome, then you convert your research question to a testable hypothesis, a null hypothesis. Then, there are more examples of this. If you're like, okay, I understand this, but I want to see a little bit more, you can have a look at the book. Then, the next thing is to choose the appropriate method. I already told it, but it boils down to this for the purpose of this lecture. You can compare means with some version of the t-test, and you can compare frequencies with some version of a chi-square test. Then, once you know what method you're going to use, that is when you can estimate the required sample size. So that means you already need your primary outcome, you already need a testable hypothesis, and then you can choose whatever method is available to test that hypothesis, and only then is it possible to give a estimate of the sample size that you need. Again, collect as many as possible within a reasonable investment of resources. Oh, sorry, there's a question. Can you express alpha and beta in the same equation to see how they cohere? Yes, but it depends on the test. So I will not do that here, but I will show what it looks like for the t-test in a couple slides. Okay, next, you want to collect as many as possible. Even if you have a lot of resources, you don't care about money, you don't care about time. I don't know to who this applies, but you never know. You still should substantiate your choice, right? Because what if you don't conclude a significant difference? Then if you have said that, okay, I want this many samples at least because that should give me enough power, if you then still don't conclude a significant difference, then your results are valuable because with a sufficiently powerful study, you didn't detect an effect. Yeah, that's a nice conclusion. Then third, you should assess the feasibility. You know, maybe you need hundreds of samples to do this, and you only have time or money or other, resources to do several dozen. Okay, here's a specific example of the sample size. This is for a 2-sample, 2-sided t-test. And for a certain simple test like this one, you can express the relationship between the type 1 error rate, the type 2 error rate, so that's coming back to his question, the variance, the effect size, and the sample size exactly. This is an exact relationship, this is not an approximation, but only for the 2-sample t-test. Yeah, 2-sample, 2-sided t-test. So what does this say? So n is the sample size, and the sample size equal to the variance, so that's the extent to which individual observations differ from each other, times how strict you are about either error rate divided by the difference in means. Yeah, so this is the critical value for alpha, this is the critical value for the power that you desire, and this is the difference in means. Okay, if this is not intuitive, let me explain why it's like this. Because even though this is only true for the two-sample, two-sided t-test, this is approximately true for anything that you can do. Because it says that if the variance is large, if the extent to which individual observations differ from each other, if that is large, then you need more samples, right? Because there will be more overlap. Then it also says, if the difference in means is very large, then it's very obvious. And you're going to divide by that. So the larger the difference, the fewer samples you need. And if the difference is very subtle, you need a big sample to actually see a significant difference. Now, then finally, if you're going to be very strict about what counts as significant, what is sufficiently powerful, then yeah, of course you need more samples because you're being more strict, right? So even though, you don't have to memorize this, but even though this relationship is quite difficult to express for even just like multiple linear regression or GLM, it's still approximately true. Yeah, just think in terms of which things are going to increase how many samples I need, and which things are going to decrease how many samples I need.
Franz?
Yes.
How are you supposed to know beforehand what the variance or the difference means?
Yes, great question. That is a really good question because you can't. That's why you're doing a study, right? To find out. But you can make a guess, right? Or you can look in literature. What did other people find for these values? Those are still estimates, but you can do that. And I think the best thing you can do when you can't make a reasonable guess and there is no literature on the subject, then you can define a minimally biologically relevant difference, right? And then, then there's still the variance. You still don't know that. You can just simulate. We'll get back to simulation in the advanced statistics course with small variance, with medium variance, with large variance, and see how does that affect my sample size. And then, you don't have to report one number to ethical committee. You can just say, okay, we simulated across a range of values, and these are realistic outcomes in terms of required sample size. So we recommend the ethical committee to approve, 100 mice, something like that. Yeah? Yes?
So, would it be possible, for example, if you have the time and resources to do so, to get a smaller sample first to determine like a baseline difference in means, or would that contaminate your other research?
Okay, let me reiterate the question for people listening. Could you first collect a small set of samples and then use those to estimate these things, the variance and the difference in means, so you can substantiate the required sample size for the actual study? Or would that contaminate the end result? It kind of depends how you do it. It's a great idea, and this is essentially what all pilot studies are meant for, right? It's a feasibility assessment of the actual study. You should definitely do this. If you have no idea, this is a great way to go about it, because also for your simulation, you can feed it with the values that you found in the pilot study. And that's a great way to go about it. The one thing that you shouldn't do is include the data from the pilot study in the final study, because you already used those random samples to estimate the effect size. And if you're going to use it in the end again, use the same data to generate multiple results, and that's going to inflate the actual estimates. Yes?
What do you mean by
Critical value, okay. Can people see this? Sorry, let me quickly check what people are seeing. Okay, I'll turn this a little bit. No? Oh, wow, it moves. Okay, is this visible? Not really. Never mind, I have a computer. What am I doing? Okay, let's draw this. Okay, what is the critical value? So the best way to explain this is to just go over what a specific test is, like the t-test or a chi-square test or whatever. So I'll just quickly go over the t-test, right? What you do in a t-test is first you define the null hypothesis, like there is no difference in means. And then we have a distribution, which I'm going to draw very sloppily now, of values that we expect from samples if the population has a real difference of 0. Yeah? So this is the, let me reiterate, and let me make it bigger. So this is supposed to be a normal distribution or a t distribution. This is the value under the null hypothesis, 0. But we are drawing a sample. It's a random subset. So there's a really good chance there's going to be some small difference, even though in the population the difference is 0. Now, this distribution, this probability distribution, defines how large the chance is that if we draw a random sample from a population without a difference, that we actually find a difference of a certain size. Now, then you choose alpha, right? You choose a bound, a threshold for the accepted chance of false positive, and that will give you a critical value. So if I choose alpha 0.05, that means 5% of this distribution over here and over here. Yeah, and now I'm going to not use red because for some reason that updates more slowly, I think due to color compression, but blue. Yeah, so this is 5% of the probability distribution, and what we're really saying is if there is no difference in the population, then only 5% of samples drawn would have this extreme a difference. And how large that value is, how large your t value should be in the case of a t-test, that's the critical value. And if you exceed it, you call it significant. Yeah? What I also recommend, if it's not quite clear yet, is to watch the video on the t-test, this one, or the video on the chi-square test, because I explained what I just explained here. I also explained in those videos to quickly go over what is this p-value, which, I forgot to say that. The p-value is the surface area, right? If we color this in, it fills x% of the distribution, and that is p. Yes?
I thought I understood that for the beta, you can't set a specific number, but how can you put it in your calculation?
Yeah, good question also. Okay, so when you go to a, again, let's say we're working with live animals because there it's most critical that you accurately define your sample size. They're going to say, okay, what is your estimated sample size? And then you're going to say, we want at least an 80% chance to find a difference if there is a difference. Or maybe you want a 90% chance, right? Because maybe this is a very difficult research, only your research group can do it. So you say, okay, let's make a count. We want a lot of power. We want a 90%, 95%, 99%. So you define it, right? Either you choose the sample size beforehand, or you say, I want at least this large a power, and then you fill all the others in, you know, the critical value for alpha, the estimated variance, estimated difference in means, and then you can get an answer.
It's the same like alpha, you can set it the way.
Well, if you haven't chosen your sample size yet, you can set it, yes. But especially for more complicated studies, you can't really set it. You can just say, okay, if we have this many samples, our power would be approximately this. So can't quite set it, except for very simple tests where there is an exact relationship. Yeah? Are there any other questions? Okay, then let's continue. Okay, so for simple tests, t-test, chi-square test, but also for ANOVA, for simple linear regression, it is still possible to express this relationship exactly. You know, the more complicated technique, the harder it gets to get an exact relationship. But there are calculators. We don't have to do it by hand. So you can search for a power calculator online. Just make sure you trust the resource. Or you can use the Power PWR package in R, which contains power calculations, which is equivalent to sample size calculation, for a variety of different tests. And they also include examples. So you can just copy the example and change the values to whatever is appropriate for your study. Yeah? Just keep in mind that even though the relationship for a simple test is exact, the numbers that you plug in are more of a guess, right? So what you should usually do is define a range of sample size that you say, okay, we think we need between this and that by filling in different numbers in those sample size calculators, slightly inflate the variance, slightly decrease the actual difference that you're going to find, and then you have the lowest chance of having an underpowered study. Okay, Yeah, just small summary. You need more observations if the difference of interest is small, if the variance is large, which is the extent to which things differ from each other. If you set the threshold very strict, you need more samples, and if you want high power, you need more samples. The other way around, you need fewer samples. So if the difference is really obvious, then you don't need many samples to see it. If the difference is very precise, there's no overlap between distributions, then you don't need many samples. And if you're more tolerant of either error rates, then you can also deal with a smaller sample size. Okay, finally, both for online calculators and for the power package in R, you will often have to fill in Cohen's d as the effect size. And this is actually a combination of the difference of interest, so here it is for a t-test, yeah, the difference in means, divided by the standard error, standard deviation. So yeah, it's a combination of the difference and the extent to which it varies to standardize it, but that you have to keep that in mind, because if you fill in the actual difference here, then the calculation is not correct. Now, this value is very popular in behavioral sciences, but yeah, there are people who write papers about how you should interpret this, but that only makes sense if you already know this research well, you've already performed this kind of research, and you have seen, you know, what is a big difference between bacteria, what is a small difference between this and that, and then you can start defining these interpretations. But I would say, if you're studying something new, this makes no sense, because what is very small for one type of research may be critically important for another type of research. So yeah, just fill in different kinds of values and see what the effect is on the required sample size. Okay, if all else fails, if you really can't come up with reasonable values, or if you did and you see that it ranges between, I need from 5 to 500 samples, that's not very useful information, then what you can still do is calculate a minimal required sample size. Yeah, unlikely to give you enough power, but then you know this is the smallest number of samples in order for me to be able to do any kind of analysis at all. Now, this is quite simple. This is the number of estimated parameters in that test or model, and you add 1. So you have 1 degree of freedom left. Now, for one sample t-test, all you do is estimate 1 group mean, so you'd need 2 samples. Yeah, for 2 sample t-tests, estimate 2 means, you need 3 samples, etc. ANOVA, there are a number of means, add 1, you have enough samples. Yeah, not enough to conclude a significant difference probably, but you do have enough to actually run the model or run the test and see the results, and you can use those results, you know, in the context of a pilot study to substantiate the sample size after that, if you're going to do a bigger research study. Yeah, so that's something that I also use a lot. Okay, I've talked about variance a lot in this lecture, which is the extent to which individual observations differ from each other. And if that extent is small, then the variance is small. And if the extent to which individual observations differ from each other is large, then you have high variance. Now, some aspects, some components of variance we can influence, some we cannot. Yeah? Variance is caused by several things. There is a natural variability to any biological process. Oh, sorry, let me read the question. You mentioned when you try to guess a model, we can first use small samples to test. Will the small sample always represent? Yeah, so yes, when you do pilot study, your sample is very small. So the question here is there not a problem that the pilot study is completely irrepresentative of the population? And of course that's true, but that's also true with a larger sample, just to a smaller extent. So we're just trying to guess a required sample size with the pilot study, and worst case scenario, you're off with the required sample size, but you're still going to use the bigger study to actually make a conclusion on the research question, right? That's not the purpose of the pilot study, it's just feasibility. So even if your pilot study is somewhat wrong, that doesn't matter for the final results. Yeah, it's just a way to estimate how many samples do I probably need to conduct this kind of research. Okay, what is causing variance? There's a natural variability to any process, right? There are small genetic differences between individuals, environmental differences, background radiation. However small it is, there are many things that we either do not or cannot measure, and things that we cannot measure with infinite precision. So there's also measurement error because of that. Because even if you're really good with a pipette, you probably cannot tell the difference between 0.9999 and 1. So however small that is, there will be an effect on the estimate. It will cause a measurement error. Now, then finally, there's sampling error, because your sample is random. So there's always a chance that your variance is different from that in the actual population. Okay, so the last one, sampling error, is basically this. You have a population, you're going to draw a random subset. Drawing it randomly will deal with any potential bias, there's no issue there, but it's still a random subset, so you can get unlucky, so to speak. Yeah? Okay, the last really important thing that sometimes goes wrong in studies is that they mistake pseudo-replicates for true replicates. So what I'm saying here is that measuring leaves from 5 plants is not the same as measuring 5 leaves from a plant, right? Because in this case, it really depends on how this particular plant was growing, how the soil was in this particular pot, how many times we watered it. Those aren't independent, they're correlated because they all belong to this one plant. And sometimes it's very obvious, like this with plants. Sometimes you're conducting a research and it's not so obvious what the experimental unit is, but you should think about it. Like, what is the smallest experimental unit that I can still apply treatments to such that it will yield me independent observations? Yeah, and everything I told in this lecture about required sample size, etc., pertains to the number of independent observations, not pseudo-replicates. Now, for those of you that will follow the advanced version of the course, one of the really cool things you can do with a mixed model is incorporate pseudo-replicates to reduce the measurement error. Yeah, so that has some small effect on power, but it doesn't affect the minimal required sample size. Yeah. So what I'm saying is you can use pseudo-replication in a study meaningfully. Like measuring, for example, the same patients at different points in time. You can still use it in a model, but the number of patients that you need is not going to be affected by how many times you measure them. Okay, small summary. You have to define a primary outcome. You should not start collecting data before you know what you're going to do with it. That's critically important. So in case you're thinking of doing a hypothesis test, convert your research question to something testable. Then find an appropriate statistical tool to actually test that hypothesis, which will give you a starting place for the required sample size. Then, before you start, try and think what could introduce a systematic difference between observations. Are there any potential source of bias? And if you do find anything, then randomize across it. So another example, we talked about 2 ovens, but it can be much simpler. Let's say 2 students are working on the same project. Don't let student A do all treatment A and student B do all treatment B, because any difference in how they measure is going to be present in the final results. So randomize it across the students in that case. Yeah? And then design your study around these 3 aspects. Okay, I've already said it several times, but it is really, really important. If you're not sure about something, talk to your supervisor. And if your supervisor is not sure, talk to a statistician. Because as Ronald Fisher, from the Fisher information, a lot of important concepts in statistics, really famous statistician, he said, to consult a statistician after an experiment is finished is often merely to conduct a post-mortem examination. He can perhaps say what the experiment died of. Yeah, so the point being, if someone comes to me and they say, hey, I have this data, can you analyze it? Then my first question is, okay, what do you want to do with it? What's the research question? And well, not most cases, of course, but very often I will have to say, okay, but you can't do that with the way you collected these samples. You can't draw that conclusion. And then there's going to be some debate over that. But, if you're trying to publish it, you'll get the same comments. So if you're not sure, ask for help with the required sample size, with how you should randomize, with how you should design your study from a statistical point of view before you collect any data. Yeah? Yeah, okay, you guys can come to us. We will try to help you. During the bachelor course, we will not really have time to also help master students, but whenever we can, we'll try to help you. If you think you just have a simple question, you can always go to Cross-Validate, which is a great resource for statistical help, and also study design questions are welcome there. And yeah, with that in mind, you're all going to do internships during your master, so good luck designing your experiments. Are there any questions? I'll check the chat quickly. I don't see anything. Okay, if not, then we'll look at the time. It is just past 10. So let's have a 13-minute break. Meet at 10:20. And then Harold will talk about his part. Yeah, for the people listening, I will now mute the stream so that you